Hello again, biologists. Mr. Kinnear here with another video lesson for National 5 Biology. We're on Unit 1, Cell Biology, and today we're looking at osmosis, which again is part of Key Area 1.2, and it's learning points D and C. So this is the third of our videos looking at Key Area 1.2, transport across cell membranes. We've looked at the structure of membranes, we've looked at diffusion. We're now going to look at osmosis today. And here's what that looks like on the SQA documentation. So at 2 points, osmosis, what is it? And also how it impacts on animal and plant cells. So in terms of learning intentions, I'm going to talk you through what osmosis is all about. Investigating osmosis, so what we can do in school, but also as well something you can do at home. And we'll then look at how animal and plant cells are affected. So what is osmosis? Well, as I've said before, it's a special case of diffusion. So as I go through this definition, if there's any aspect of the definition missing, then it's not osmosis. It may just be diffusion. What we need to remember though, is that every single one of the cell types that we discussed in Key Area 1.1 is surrounded by a cell membrane. And as we looked at in Key Area 1.2, the structure of that membrane, it consists of a phospholipid bilayer with proteins embedded in it. And it's semi-permeable, which means some molecules can pass through really easily and some cannot. Either side of that membrane will be water, and that will be of varying different concentrations because it will contain salts, sugars, other nutrients that are essential for life. That means that the water concentration on either side of that membrane may be different. Because osmosis involves the movement of water molecules. And if it's not water, it's not osmosis. That's from an area of high water concentration to an area of low water concentration. That's through a selectively permeable membrane. Now, I've got to be clear here, it's high water concentration and low water concentration. Now, if you're sat there watching this video with a glass of water, if you poured it from the tap, then it's not 100% water. It's very high water concentration, but there will be salts and other minerals in that, even if it's a bottled water. But it will be very high water concentration. You could lower that water concentration by adding sugar or salt to it. Again, it'd be the same volume of water. Because you've added something else into it, then you're actually reducing the water concentration. Wouldn't make it particularly drinkable. But likewise, if you added diluting juice to your glass of water, equally, you are reducing the water concentration because some of the fluid in that glass now is something else, not just water. And that's important when it comes to the aspect of osmosis. And another thing we need to remember is that osmosis, like other forms of diffusion, is a passive process. It does not require energy. It's going down that concentration gradient, high water concentration to low water concentration. What we're going to do is look at an animation that shows this process with two different solutions either side of a selectively permeable membrane, and we're going to see what happens. So in this animation we have water in the blue circles, sugar molecules in gold, and as you can see the sugar molecules are too big to pass through our membrane. Water molecules are moving back and forward all the time, but there is a net movement. It's a net movement from left to right, as you can see from the numbers. And what's happening is that they're trying to get to an equilibrium. So we're trying to balance out the water concentration on either side. And that is going from left-hand side, high water concentration, to low water concentration on the right-hand side until we get to an equilibrium. So to give you an example of an osmosis investigation, quite a simple one we can do in the lab, and it's something that comes up within questions and SQA papers. We take cylinders of potato, roughly around the same size, measure and record the mass of each. You then place each cylinder into different solutions, one of pure distilled water and one within a strong salt solution. You'd leave them overnight. You'd then take them out and measure and record the mass. And let's look at what would happen. So we'll look at the potato in pure water first. So what we have is high water concentration, often shortened to HWC, in the test tube surrounding our potato cylinder. And within our potato cells, we'll have a low water concentration, often shortened to LWC. So what's going to happen? The water molecules are going to move from high water concentration in the tube to low water concentration in the potato cells. That means the potato cells are going to gain mass, they're going to gain water and get heavier. So the potato cylinder will be heavier at the end of this experiment. Now with our potato in strong salt solution, we've got the same volume of water surrounding our potato, but what we've done by adding lots of salt is we've lowered the water concentration. So that then turns the tables. We haven't changed what's inside the potato, it's the same potato, but we've now changed the concentration gradient. So we've got effectively low water concentration in the tube and a higher water concentration in the potato cells. Therefore, the water molecules moved from high water concentration inside the potato this time to the low water concentration in the test tube. That means that our potato cells lose water and the potato cylinder gets lighter. So we demonstrate that concentration gradient and the movement of water because in both instances there's a change in the mass of the potato. In pure water it gets heavier, in the strong salt solution it gets lighter. So if you want to jazz up your experiment a little, instead of using potato, you can use gummy bears, get them from your Haribo Star Mix. You can simply make up salt solution, sugar solution and plain water, leave them in at least overnight, minimum 8 hours, and then obviously compare the bear size to a control bear, one that you haven't placed in any of the solutions. Again, because of osmosis, you will see a difference in the sizes, and that comes down to water molecules moving either in or out of the bear from high water concentration to low water concentration. So that's something to help you visualize the process of osmosis. So it's all very well looking at gummy bears and lumps of potato, but we're now going to drill down into looking at what happens to the cells. And again, remember that our plant and animal cells are surrounded by a semi-permeable membrane that allows water and other small molecules to diffuse across. So there we have an example of a generic plant cell, and this time a specialised cell from humans, a red blood cell, both surrounded by a plasma or cell membrane. The additional thing is that plant cells have a strong cell wall surrounding the membrane which offers support and protection. And that's going to be key to what happens with osmosis between plant cells and animal cells. So we'll start the process with an animal cell. This is a red blood cell. If we take that red blood cell and stick it into an area of low water concentration, so for instance into salt water, then we know that water will move from high to low, so therefore out of the cell. That cell will then shrink and become shriveled. It may prevent it doing its job within the body. Equally harmful could be sticking it into an area of high water concentration, so pure water. Again, the water will move from high to low, so into the cell this time, and that may cause the cell to swell and burst. Again, equally damaging. If it's stuck in an isotonic solution, so equal water concentration either side, there's no net movement of water across that cell membrane. Therefore, the red blood cell stays as it is. Next up is our plant cell. So as we know, it has a cell wall, so some of the problems that our animal cell, our red blood cell, faced might not be the same. So this time we'll start with looking at high water concentration, so again pure water, and again we know that the water will move from high to low, so into the cell, but with that cell wall protecting it, it's not going to burst, but it does become swollen and firmer, and therefore the word we're looking for there is turgid. So if a plant cell is placed in pure water, it will swell and become turgid. On the opposite side, if we take that plant cell and stick it in a low water concentration, so salt solution, we know again that water will leave the cell. However, that plant cell wall, providing it that support, the cell inside the cell wall may shrink. It won't become shriveled because that cell wall will still provide that rigid structure. That therefore is termed to be plasmalized. So the internal structures, the vacuole, the cytoplasm may shrink away and pull away from the cell wall. The cell wall still manages to keep the structure of the plant cell intact but that therefore is referred to as plasmolysed. So that's us for today. We have covered off area 1.2D and 1.2E. So we've clearly stated that osmosis is the movement of water molecules from high water concentration to low water concentration through a selectively permeable membrane. We've also discussed how animal cells can burst or shrink, plant cells become turgid or plasmolysed due to the different concentrations that we might place them in. So those are key things that you guys need to know. So from here, what we'd like you to do is use this information, use Scholar, use Bite Size to create your own notes regarding osmosis, making sure you have a clear definition in your jotters, and also then diagrams of what happens to our animal and plant cells in high water concentration and low water concentration. So those are really important things for you today. So that's us. Thank you very much for listening, and we'll see you again soon. Bye. And that completes our learning from today. Today we've looked at osmosis. It's the movement of water from high water concentration to low water concentration across a semi-permeable membrane. And as it's a passive process, it doesn't require any energy. There are simple experiments which can show the impact of osmosis, and these experiments often come up in SQA papers. What also comes up is the effect that osmosis can have on animal cells and plant cells. So animal cells which can burst or shrink due to the movement of water, plant cells which can be turgid or plasmolysed because of osmosis. And that then is down to the presence of a cell wall on plant cells and not on animal cells. So hopefully you found that informative. As always, if you have any problems, questions or queries, give me a shout and I'll speak to you again soon. Bye.